I think I think that's everything as far as health uh, housekeeping. What I'll do is I'll hand it over to Eric Page, who's going to introduce our lightning talk speakers and talk a little bit about the special issue that we had with the Canadian Journal of Plant Science.
Okay. Thanks, Rob, for the introduction. Welcome, everybody, to the 2024-25 CWSS AGM. We thought we'd offer a little bit of content just to get everybody ready for February and Vancouver, our joint meeting with WSSA, which I know we're all looking forward to, visiting David and Jichul in Vancouver. So with that in mind, we thought we'd give you a little sampling of what has been some work that Jichul, myself, and Charles have been doing over the last year, which is basically co-editing a special issue in the Canadian Journal of Plant Science. So CJPS is our sponsoring journal for the Canadian Weed Science Society. And so this has been an endeavor that we've undertaken over the last year. So I'm going to share my slides, and I'm going to talk a little bit about our goals of the special issue, what was produced out of it, and then I'm going to invite our three speakers to give lightning talks. So again, brief overviews of their publications, which are now available open access through the CJPS website, and they'll be able to sort of introduce you to their publications through these lightning talks. So again, we'll go 5 minutes or so, 5 to 7 minutes for the speakers. If there are any questions, I would encourage everybody just to put your hand up. So that's available down through the reactions option down on the bottom of your window. And then between Jichul and Rob and I, we'll do our best to provide you a platform and curate those questions for the speakers. Failing that, please feel free to just drop questions into the chat and we'll do our best to bring them up for the speaker. So one thing to bear in mind too, when the speakers have their presentations up, they can't always see your questions if you put them into the chat. So if they're not responding, feel free to unmute and get our attention, and we'll be happy to give you the platform for asking your question. So I'll share my screen here and we'll get started. Okay, so can everyone see my screen? Yeah, Jichul?
It's not a presentation mode.
How's
There
that?
we go. Yep, that's good.
Yeah, there we go. Okay, so as I mentioned, we had a special issue recently with the Canadian Journal of Plant Science, and so the focus of this special issue was forging new paths to manage herbicide-resistant weeds. And so again, Charles and Jichul were my co-editors on this, and this is with CJPS. And one of the reasons we wanted to focus on this, so it was a topical choice of the co-editors here in terms of the focus of the issue, and really just wanting to bring new light to the issues around herbicide resistance. Obviously, this is not a new topic to any of us working in the field of weed science, but some of the numbers recently that have come out of some recent Canadian literature on the impact of herbicide resistance just demonstrate the scale of the challenge that we all face in this industry. So just highlighting in particular, herbicide-resistant weed costs for farmers on the Canadian prairies were estimated by Hugh Becky, a former member and president of the society, to be about $530, not $530, $530 million annually. So just giving you a sense for the cost and the scale of the issue that face farmers on the prairies. Regionally here in Ontario, Dr. Sikma has produced some excellent numbers in terms of the impact. He estimated that in Ontario, it's about $43 million annually with the cost of yield losses and resprays associated with managing herbicide-resistant populations. So herbicide resistance as a challenge is not new. We wanted to bring a different lens to the issue of herbicide resistance. And in particular, this special issue was focused around the idea of biovigilance and biovigilance as a framework for addressing herbicide resistance. So what is biovigilance? This is the figure that Charles produced for the special issue, just sort of highlighting where all the submissions that we received place throughout the biovigilance continuum. So we had 12 submissions, And what this really just goes to show is that we were fortunate enough to receive submissions from across the various components of biovigilance. So as a conceptual framework, biovigilance is put forward to help address new threats to farming operations by placing continual research efforts along this continuum of awareness, detection, assessment, understanding, and then the appropriateness of the actions that are being undertaken to manage the herbicide-resistant issues. In and of itself, biovigilance is defined as the study of unintentional effects of climate change, pest introduction, and new agricultural production and farming practices on pest populations. And so what we hope to do in focusing the special issue around these topics was really to try to reframe that concept of, how you address herbicide resistance, weed populations. Is it from, a raising of awareness, which we're going to hear from Dr. Sean Sharp today about the threat that Palmer amaranth represents for Canadian agricultural systems. So bringing awareness of that potential challenge. Is it detection? We're going to hear from Eric Patterson today about early detection and the technologies that we have available now to identify herbicide-resistant biotypes. Is it about mitigation or appropriateness of the response to it? And we're also going to hear from Andrew Mackenzie-Gopsel on his harvest weed seed control. So all of these things fit together into this special issue. With that, sorry, I wanted to highlight this before I get too far on. The QR code down on the right-hand side will take you in a direct link to the landing page for the special issue if you're interested in reading the literature that's being presented to you today. So I just encourage you to take a quick snapshot of that. Otherwise, we will put a link on the Canadian Weed Science website so that everybody can access this special issue. I'm going to go in reverse order in terms of introducing our speakers and then end on our first speaker. As I mentioned, we're going to have Dr. Andrew Mackenzie-Gopsel. Many of you will know Andrew through his excellent work out on PEI in terms of their potato production systems and weed control out there. He's going to be talking about harvest weed seed control in potato systems, which is a rather unique topic. And so he'll be talking about the potato vine crusher as a mechanism for managing weed populations there. Andrew will be our last speaker. Prior to Andrew, we're going to have Dr. Sean Sharp. One of these three pictures is Palmer amaranth. One of them is Sean Sharp. I'll let you guess which one is which. Sean's going to be here talking about water hemp and Palmer amaranth biovigilance. Again, raising awareness of the challenges that they pose for agricultural production in Canada. And without further ado, our adopted Canadian, a man who is north of me today, Dr. Eric Patterson is joining us from Michigan State University. We love having Eric at our meetings. It's always great to have that contribution from him and from Greg and Hillary. We love having Americans at our meetings. So I'm going to stop rambling now, and I'm going to give the floor over to Dr. Patterson. Thank you for listening, and I'll be back to help moderate some of the questions.
Thanks, Eric. I wish I could call myself a Canadian, at least after our last election. All right. Thanks so much for the invitation. Happy to talk about this work that my graduate student, Giuliano, was working on. And it was a really fortuitous alignment of this special issue with his work. So here in Michigan, we have a plant and pest diagnostics clinic, and we're unique, at least in the United States, as having a full-time weed diagnostician. So a lot of our plant and pest diagnostic clinics are staffed with pathologists or maybe entomologists, but we have a weed scientist in Erin Hill. And so when I got here, I talked to Erin Hill quite a bit about her diagnostics, and she is one of the few people offering herbicide resistance diagnostics in the country. Talking to her through her work I was thinking about ways to make it more efficient for her to do her work. So Michigan farmers were consistently having problems with group 2 and group 9 herbicides, so ALS inhibitors and EPSPS inhibitors. And for each population that was sent in, we were running whole plant diagnostics, dose response curves, and for these 4 species in particular. So you'll recognize them, hopefully, as water hemp, ragweed, mare's tail, and Palmer amaranth. So really our goal was to reduce the cost and time inputs for these really repetitive assays that she's just running consistently. And the good thing about group 2 and group 9 resistance is we know a lot about the most common resistance mechanisms to these groups, right? So there's a lot of target site resistance for these. And target site resistance is great because it's very easy to detect with molecular assays. Why would we want to use a molecular assay? So obviously doing whole plant diagnostics takes a long time. Growers usually at the end of the season are collecting seeds, sending them to us in September, October, November. We have to break dormancy, grow the plants, spray the plants, evaluate the plants, and get them a report sometime in February, March, right before the next planting season. Theoretically, if you know the mutation, you can skip all of that and just sequence the gene directly out of the seed or out of a seedling, something like that. And if you know that this SNP causes resistance, then you can report back to the farmer pretty much immediately. Theoretically, you could even report back within season if the grower is willing to send you tissue, let's say in June, after the application fails, you could tell them pretty much immediately. So we had this goal of, for these really common diagnostics, of reducing it down to just a couple of weeks between a farmer seeing a problem and us getting them at least some information about what's going on with these common issues, right? For target site resistance, there's a lot of mutations in ALS that can cause resistance. They're all kind of laid out here. At the top, you have your known SNPs in any weed species. And then in the bottom 4 rows are the SNPs that are discovered in the 4 species we're talking about. So there's 3 identified in common ragweed, 6 in palmer, 2 in water hemp, and 4 reported in horseweed. Similar story for EPSPS. It's really this proline 106 mutation that we need to survey for, or possibly the TIPS mutation, which includes the threonine and proline at the same time. So we need to sequence one region in EPSPS and essentially the whole gene in ALS. Now, these are known SNPs. The problem is when you look through the literature, a lot of the primers that people develop for these are specific to their population and may not be universally robust to all palmer or all ragweed. So that was another major objective of our work, is to make primers that would work on any sample from all of Michigan, essentially. So we started off with a bunch of resistant and susceptible populations that we had in hand. And we just did a ton of work developing primers, optimizing temperatures, optimizing DNA extraction techniques to make it very reliable to run on these 4 species. And then we followed that with a field assay where we sampled, 100 plants from the field from these 4 species and just double checked that it worked no matter what plant and any condition that we could get our hands on. This is a breakdown of the primers that we designed for ALS sequencing. You can see these are the ones that we found to be the most universal. We started off with primers in different locations, and these are the survivors, essentially, right? Because we wanted that 100% always working assays. Essentially, we do DNA extraction, we do a PCR amplification, we send that for nanopore sequencing rather than Sanger because it's a little bit easier to interpret and analyze, and it's cheaper for us now than Sanger was. And then we compare it to our susceptible sequence, pretty standard SNP genotyping protocol. For instance, here's a proline 197. You see the susceptible sequence on top, the resistant sequence on bottom. We can then pretty confidently report to the grower, yeah, that's a resistant individual. Okay. Just a quick results. What did we find when we sequenced a bunch of Palmer amaranth from around our resistant populations? We found a lot of these proline 197 mutations, including the heterozygote. We even found the asparagine 376 and the tryptophan 574. And we can see in our populations that they're segregating for these SNPs. And in some populations, we actually find multiple of these SNPs coexisting from the same field. So we have lots of interacting SNPs all within the same populations. is interesting. Water hemp, it's really this proline 197 mutation and this TRP 574 mutation, either in the homozygous or heterozygous state. And you can see we have lots of resistance in water hemp to ALS inhibitors. Common ragweed, proline 197, we get this interesting alanine 205 mutation as well, and the proline and the tryptophan 574. Less ALS resistance in our greenhouse populations for this. And what was cool for horseweed is we only have the proline 197, and when we have it in the entire population homozygous. This is probably because horseweed is a selfing and not a outcrossing species as opposed to the other weeds we showed. Same in the field. We can use this for any field population. We actually found less resistance than we thought we would. Everyone says, oh, it's just ubiquitous, right? But there's actually quite a few susceptible individuals. But once you reach 10 or 20% of individuals, farmers notice those, and we call it a resistant population, even though they're controlling probably 90% of individuals, 70% of their individuals still. I'm going very quickly. You can read the paper for more details, I guess. With EPSPS, again, we're looking for those SNPs, at least in horseweed. But in the amaranthus, the more common resistance mechanism is copy number variation of the target site, which again is easily diagnosable with genotyping. So essentially, we do the same thing for mare's tail. We just extract the DNA, sequence that proline 106. And for the amaranthus, we do quantitative PCR directly off of gene amplification, DNA. And you can see if they come up together, that means we have the same number of copies of EPSPS and ALS. But if they come up at different cycles, if EPSPS is earlier, there's more copies of it in the template. So really easy to diagnose in the lab, not very expensive. What did we find? What was really interesting was even though non-target site resistance is commonly attributed to horseweed, anytime we had a resistant plant, we found the proline 106 mutation. And we attribute that to, now that Martin Laforet did the genome, there's actually 3 copies of the EPSPS gene in most Asteraceae. And we're not sure that previous literature was always sequencing the proline 106 from EPSPS2, which is the functional copy in mare's tail, and they weren't amplifying a pseudogene of it. So now that we look for it and we redesign primers to specifically look at the correct EPSPS, we almost always find it when we have a resistant plant. That's not to say we don't find some NTSR, but we're finding this in almost all of our resistant populations, which was good to know. In water hemp and Palmer amaranth, when we had resistance, we had copy number increase. Of course, hundreds of copies in Palmer, but between 4 and 10 in our water hemp populations. What was really important here is that we confirmed that ALS is a better reference gene. Other people were trying to move to beta tubulin as a reference gene. However, if you look at the genome, beta tubulin, there's multiple copies, and the primers actually underestimate the amount of EPSPS copies because there's too many reference copies of beta tubulin in the amaranthus. It's actually better to use the ALS gene to get a 1 copy reference. So we showed that very nicely. And then in the field, it works gangbusters to detect resistance as well. So whether it's mare's tail or amaranthus, we're easily able to detect resistant and susceptible individuals from the field. Conclusions. Right, so diversity and frequency of resistance varied between species, especially in the cross-pollinated species. We get a lot more heterozygosity. We've optimized these primers. They should work on most plants unless you get a lot of sequence diversity, and they're pretty efficient at detecting resistance. ALS is a good housekeeper for copy number variation in the amaranths. And for Michigan growers at least, we've reduced for these herbicides from a 9 to 10 month turnaround to a 3 or 4 week turnaround, which we're really happy about. And it's cheaper just because of the time spent doing it. And we've implemented this as a service, so people can send us plants and we will do that for them pretty much for a nominal fee, maybe $10 a plant, something like that. Okay, I just want to acknowledge my students who worked on this, mostly Juliana Solsbeck standing here, who's now a PhD at Oregon State. So I know I probably went over time, but thank you for allowing me to rush through that. And I'd be happy to answer questions if there's time.
Awesome. Thank you very much for that, Eric. I'm amazed you guys have the time to be able to do that as a service provider for the farmers, but that's great. Is there, I know what's happened here with some of the tests is that they've been kind of, the IP has been through agreement sent out to commercial labs. Is that part of your plan going forward, or is it going to be an MSU in-house type of thing?
So we would be happy to give those to any diagnostic clinic that wants to use them. These aren't protected by any IP or anything like that. The techniques are pretty much universally available because it's just a typical PCR and sequencing event. If you know how to do PCR, you should be able to run it. So it's not protected.
Cool. I'm seeing some questions here in the chat, so I'll just repeat them for you here. So Hillary Sandler, who's listening, she's asking how many samples do you process through the diagnostics lab?
So this is relatively new. So the paper came out in 2024. We haven't widely advertised it. So we've probably run about 300 plants from around Michigan last year. And again, we get a kind of a bulk discount. So doing 12 at a time is cheaper than doing 1 at a time per plant. So if we can do all of our DNA extractions together, that's what we do.
Cool.
So I imagine if you scaled it up with robotics and some like in-house nanopore sequencing, you can make it very cheap, but we're just not really interested in that level of scale yet.
Right.
Yeah. Maybe for when people are testing for 2,4-D resistance in dicamba and they're doing it as a service, you would want to do something like that. Yeah.
Another question here from Francois. He's asking, what's the workflow if a sample escaped the herbicide in the field, but the molecular test comes up negative?
Right. So that's the worst case scenario, right? The problem with the molecular diagnostics is we can only prove a positive, but never prove the negative, right? So we only say we confirm resistance, not confirm susceptibility. We just assume that there's a mechanism we're not detecting. So an NTSR mechanism or something like that. So we report back to the farmer and say, no obvious, signs of target site resistance. We'd have to go out, collect the plant, bring it in, get seeds, and do the whole workup, right? So then it would go into traditional diagnostics and we'd have a 9-month turnaround.
Right, yeah. Lots of greenhouse work associated with that.
Right, yeah, and time is the biggest thing for us is time, just paying someone $20 an hour to grow plants, right? And then the process and spray them is expensive. So, we say, oh, it's $9 a sequencing reaction, but it saves you months of growing plants. Well, that's
right.
Yeah.
That's understood. No, that's excellent. Do we got any other questions for Dr. Patterson while we still have him? You guys are welcome to unmute or throw up a hand if you have a question. I'm just reading what's in the chat, and I'm just looking now to see if there's any hands up.
I'm trying to get my screen back.
Yeah, I know it's hard once you've been presenting, eh? Presentation mode. Okay, well, you were right on time, Eric. So that's why I'm being patient here for questions.
Please send me more questions. Send us samples if you want to talk about these modes of actions or others. Happy to collaborate.
And I trust maybe we'll see some of your work in Vancouver at the joint meeting.
Yes, and we're moving on. We're doing lots in PPO for the deletion. So PPO resistance and a couple of dicamba and 2,4-D resistance in kochia. So.
Cool. Awesome.
Right.
Well, thank you very much, Eric. We really appreciate you joining us. I will now ask Dr. Mackenzie Gopsel, who's the next speaker. Sorry, no, I'm going to get ahead of that. It probably just scared Andrew. It's Sean. It is Sean who's going to be I messed myself up by putting them in backwards order. Sean Sharp, Dr. Sean Sharp from Saskatoon. I'm sure it's lovely and sunny in Saskatoon and about -40. He'll be happy to talk to us about Palmer freezing on the Canadian prairies.
Hey, good morning. Am I coming through okay there, Eric? You can see my screen.
Yeah, you're good, Sean.
If I can find where my stuff went. Here it is. All right, good morning, everyone. So for those who don't know me, I spent 6 years at UF, the University of Florida. I was studying in horticulture and weed science, and I thought, I'm glad I don't have to work with Palmer amaranth. I watched all of my colleagues and grad students down there trying to combat this, and there was a lot of presentations, especially at SWSS, but a third of the talks were on Palmer amaranth, and I'm a bit surprised that I'm here now in Canada talking about this, but I think it's a very important issue that's coming our way. And so I tapped a couple of people on the shoulder for this, Sarah Martin, Eric Page, and Charles, to try to get some help. This was largely off the side of our desk to try to build some awareness. So Eric already touched on what is biovigilance earlier, so I'm just going to briefly discuss this. So this is a sort of an Ag Canada directed system, I guess you could say, inspired off of a French system for doing research for helping to manage pests and environmental considerations when growing crops. And then it is a spectrum. I'm going to turn on my laser pointer here. So it kind of does move this way, but once you get to this appropriateness step, it kind of bursts out in every direction, because your appropriateness obviously is going to feed back into mitigation, but you might go back into assessment. So this is definitely a continuum, and then it kind of flows oddly at this last step, and it does kind of flow into all of these. And then for Palmer amaranth, what I'm going to discuss today is going to be its initial history. I have a very strong interest in kind of ethnobotany, especially in ancient times, and understanding why weeds are where they are and why they are concerns in previous cultures, and how we are inheriting some of those issues. We'll talk about resistance, of course, and then in the paper, I'm not going to touch on it much today since I've only got a few 5 minutes, but there's also sections to talk about potential hybrids, which is a very concerning element for gene enrichment moving into Canada of Palmer amaranth water hemp, as well as its population dynamics. So these are the two beasts that we're concerned about. Obviously, there's water hemp in Quebec and Ontario, and then here's Palmer amaranth on the left. And then one of its identifying features is going to be these large bracts on the female flowers. And of course, both these species are hairless. On the stems, they used to be a pot herb that was boiled by Indigenous peoples. And on Palmer amaranth, of course, the petiole on older tissue is going to be longer than the leaf itself. So this is a map from Farmers and Their Languages from 2003. This is a reconstruction based on language and archaeology, to look at the origins of agriculture kind of after the last ice age, which would have been around the same time as development of the Fertile Crescent in the Far East, in the Levant. And of course, this is an area where we get our grains, our wheats, our barleys, our oats, as well as a lot of our pulse crops that we're growing in Canada, in the prairies. And then over here in this little section, this is the northern reaches of the central Mexico epicenter, where the green amaranths were domesticated, quinoa, and of course there was one up a little farther as well, where we get sunflowers. Now, the Palmer amaranth that we see in the record typically shows up right around this area here, around Baja California, and in this Corona Desert. So then in the paper, I do kind of speculate and that while it is definitely a weed, it may have either been previously a grain amaranth that has escaped, or it could have been a grain mimic that was in the grain amaranths. And then we know, of course, due to colonization, a lot of our records in this area were completely destroyed, and the systematic agriculture of grain amaranths was outlawed by the church. So, and what would happen, unfortunately, in those times is if you were caught with a grain amaranth after colonization, you would lose a hand. So there was a strong emphasis on for them not to grow these amaranths and to destroy them where they were found. So when the historical record picks up with the herbariums, we kind of see them here. By 1955, so it was about the early 1900s for the herbarium records. By 1955, Palmer is kind of spread from this region here with some waifs over here. It's kind of all the way down here. It's overlapping ranges with a few different species here. And the big part that I want to bring out here is that it's already being found around half the time in the herbarium record in artificial habitats, irrigation canals and roadsides, ditches, those sorts of things. So this is a custom map that I developed, and I detailed these out in the paper, but I find it's a little easier to present. These are state level initial herbicide resistant reports between 1989 and 1999. This is my station up here in Saskatchewan. It's -20 today, so we are frozen tight. So then you can see here we have these initial cases, but when you jump ahead to 2023, we see this proliferation of new cases, and these could either be due to novel rises in resistance or the spread of a herbicide resistant biotype. It's very challenging to pull those apart in the literature. So the blue means it's one mode of action, the darker red means two modes of action, and then these spots here, when I put this together, these are 5 to 6 way resistant biotypes. And then, so then we also talk about the aspects of its spread, and it spreads very heavily in ducks. And there was a study by Farmer out of Arkansas, I believe it was, with Jason Noseworthy, and that paper has shown that these northern populations can spread well into Canada via waterfowl when they're moving northward. And then part of the understanding stuff for this is its HR status so that we can still use chemicals. Of course, chemicals will work. If we are able to understand which chemicals will not work, we can pick herbicides that may still be effective. But of course, with Palmer amaranth, it has a variety of mechanisms, including metabolism, that helps it to exist in fields where we're spraying some of the like the group 3s, for example, that we started to, those ones take longer for resistance to evolve. And then, of course, with water hemp as well, we have many different modes of action. So getting a species test on any fields where we have suspicious pigweed, and then getting an HR test as well would be very highly recommended. And I think that's it. I cut that down quite a bit. So I'm happy to take any questions if there's any.
Thanks, Sean. Certainly some tidbits in there that I wasn't even really aware of. I like the hand loss comment. That seems a bit extreme for possession of amaranth species, lose a hand.
Yeah, and it was due to their use in the culture and humans sacrifice and those sorts of things. That was likely a misunderstanding by the church. And of course, then there was extreme policy. So policy can be effective, but may not want it to be that effective.
We certainly won't go there with government policy.
No, this is why I didn't
it could
include
be very
it
effective.
in the paper. It's more of a tidbit that I like to tell with the story. I didn't want to be the one to start putting that forward. Yeah.
Yeah, no briefing notes to the minister, please.
No.
Anyway, do we have any questions for Sean in respect to his wonderful review paper? Maybe I'll just ask a question to get it started. So are you aware of recent reports of Palmer in Canada? What's the status?
Yeah, so there was detections in Manitoba as well as in Ontario. So we're kind of seeing it around the Great Lake regions, and of course, this is areas where there's water hemp as well. So um, those are kind of be the areas where I would recommend the most efforts for to build awareness of these pests. But of course, it could come in anywhere since it spreads many, many different ways.
Yeah. And there we go. We got a question from our next speaker.
Mhm.
Andrew, go ahead.
Sure, it's less a question, more a comment. I just the you mentioned about waterfowl moving amaranth seeds around. Um, that's I there's a passage in Origin of Species where Darwin talks about doing an experiment of literally that of moving plants within waterfowl within their feathers, and I think he just moved them from different ponds and just tracked like dispersal around there. So just interesting to like whenever you can pull it back to Darwin.
Mhm.
So I see, just expanding on that, David Clements is asking, how is it spread via ducks, Sean?
So in the study they did, they destructively harvested ducks that were hunted by people in the community. So it seems like it was being eaten in the fields maybe before they start their migration, and then they actually harvested out of their guts and then counted it. And then they did a study where they took the excrement and then had a look if it would germinate through that way too. So it was definitely a very messy study. I'm sure there was a lot of paperwork with all those.
Poor grad student, we can all
Yeah.
say. Yeah, Marie-José has a question for you. Marie-José?
Yeah, I'm just wondering, like, is this water hemp, like, is it gonna move as fast or faster than this Palmer? Is it gonna move faster than water hemp, or like, I wonder what the dynamics will be, or and what'll be, is it more genetically diverse so it could even outperform the water hemp and then get higher into Canada? Like, just wondering.
Yeah, there was one paper where they did a climax Dimax model, and they had a hard time explaining its original invasion. It wasn't due to climate variables. They thought it was due to agricultural spread. And then they were saying that its movement northward was likely accelerated due to range overlap with water hemp. So there will be some competition over niches, but I think the hybridization potential will give it more adaptability as it moves northward. So I think it'll probably be accelerated by it more than hindered by water hemp. Yeah.
Sorry, just maybe I'll clarify on that last point. You mentioned hybridization potential. So what does water hemp, or sorry, what does Palmer hybridize with?
There was a range of species. Sarah Martin did the heavy lifting on that. I will refer back to the paper, but she's got a whole list of different species. Like for the ones I'm concerned about here in the prairies is like spiny and
Yeah.
red root pigweed, but for different locations, obviously there'd be different weeds of concern.
I just to bring to everybody's attention down in the chat, hearing from Jeanette there that they have single detects of Palmer amaranth in Manitoba, supported by the movement of waterfowl. I presume that's this year what Jeanette is referencing. Okay, excellent. Do we have any other questions for Sean while we have him up on the screen? No. Okay. Well, with that, thank you, Sean. Appreciate your presentation. And I will get it right this time because there's only one more choice. Our next presentation will be from Dr. Andrew Mackenzie Gopsel, who will be here talking to us about, really, Brienne's not here, and she is the queen of harvest weed seed control. So Andrew, I think you're dethroning her today. He'll be here to talk to us about his potato vine crusher. So Andrew, the floor is yours.
Can all see it?
There it is.
Yep. Okay. Oops. Okay. Yeah, so I'm going to talk to you guys a little bit today about the potato vine crusher, which we see as a new tool for harvest weed seed control. Um, so I work out in Atlantic Canada, in Prince Edward Island, and probably no surprise to you, but Atlantic Canada grows a heck of a lot of potatoes. Potatoes are the most valuable horticulture crop to Canada, with an annual value of about $1.2 billion. And of that, almost between 35 and 40% of the acreage in any given year is seeded in Atlantic Canada, primarily in Prince Edward Island and New Brunswick. And potatoes are significant drivers of our economies, not only fresh market and processing, but also bringing a lot of tourist dollars here to our region. And potatoes are not very strong competitors with weeds, and unchecked weed growth can reduce yields by upwards of 44%. And like most crops in any kind of intensive agriculture system, we have rising incidence of herbicide resistance. So here, this is just a survey we did a couple of years ago in some of the major potato producing areas of New Brunswick and Prince Edward Island. Each one of those squares on that map is an individual field or population. The green ones are susceptible and the red ones are resistant. And we found that about half of fields that we surveyed have a resistant weed. Those are all common lambs quarters, or have historically all been common lambs quarters, all with the common serine 264 glycine mutation, giving high levels of resistance to triazines. The number one herbicide that they use in potatoes in our area would be metribuzin, so really pretty high levels of resistance to metribuzin in these populations. We're also finding increasingly or more and more cases of red root pigweed resistant to metribuzin. And then just last year, we found a population of red root pigweed with a different mutation, the valine isoleucine, which gave high levels of resistance to both linearon and metribuzin. So in terms of herbicide use in potatoes, it really is those two photosystem 2 products that are used in our region. They form the foundation of like 95% of producers' weed management practices. So we're really losing all of our tools in our toolbox in our area. Now, if you've ever seen a potato harvest, it's a lot different than anything that's going to be combined. Like most horticulture crops, this is custom harvested. We have a custom harvester. And in terms of potato, we grow them in hills, and this harvester goes along, scoops up that whole hill, all the biomass that's there, weeds, potatoes, everything that's there, and then sends it up through this conveyor system. Then those brushes at the top spit all the potatoes out to the side, and then everything else gets spat out the back of the harvester. So all any rocks, some soil, as well as any plant biomass that's there. So potato vines as well as weeds. Now, looking at this, this is one of our weedy check plots during harvest. Believe it or not, there are potatoes in there. But going through this plot, you can see like a whole bunch of weed biomass exiting the back of that harvester. So when we look at that as a weed scientist, you see there might be some opportunity there for harvest weed seed control. So if you're not familiar with harvest weed seed control, it's this kind of new paradigm in weed science around, with the idea being that weed seeds can be either removed from fields or directly devitalized in fields. Because when we're harvesting a lot of our crops, we're basically seeding out weed seeds just continuously. You're probably familiar with systems like the rolling cage mill systems, such as the Harrington seed destructor or the Redicop. And these are systems that really grind and pulverize those weed seeds before they end up spreading back onto the field. But all of those systems for devitalization of weed seeds in field are limited to combine harvested crops. So when we move to horticulture crops, you can't combine harvest them. They're all custom harvested. So any of these systems are not going to be able to be used in like a crop like potato. So I was given a presentation just at our local university here, and the technician from our entomology program happened to be in the audience, and I started to talk a little bit about harvest weed seed control. And she mentioned that they had developed a system about 10 years previously to crush potato vines at harvest for control of European corn borer. So they called this the potato vine crusher, and what it did was as those vines were passing out the back of the harvester, it would pass through these two sets of rollers, crush the potato vine, and provided between 90 and 100% control of that European corn borer larvae. I mentioned it is mounted to the rear of a harvester. And unfortunately, although the system was highly, highly effective, within about 2 years of our entomologist, Christine Nerona, developing the system, new insecticides came on the market that were registered in potato and kind of eliminated the European corn borer issue in potato. So no real the system ended up not really being uptaken by any producers in our area, and it ended up sitting in the top of our barn for about 10 years until we pulled it out. And so we got it down, stuck it on a pallet, and asked the question, can the potato vine crusher be used to destroy seeds of problematic weeds in potato production systems? So we looked at this, our vine crusher, we said, what are the things that we could play with on here to try to optimize our different settings? So we narrowed it into evaluating roller speed, so how fast those rollers are actually moving, and they both rotate in on each other, as well as we measured, or we adjusted the spring tension, so how tight those springs were pulled together, which adjusted the distance and the space between the rollers. So as you increased or decreased space, you would increase pressure applied to the seeds from those rollers. To give you a little video of what we were doing, here's the vine crusher in action. You'll see my grad student just pour 1,000 weed seeds through the center of that vine crusher, and then they were captured in that bin below. And then we took that and we either did a germination assay in a Petri plate or we did an emergence assay in field soil. Won't get into the specifics of our optimization. You can read the paper to hear about that, but we did test our optimized settings on 5 different species in controlled conditions and got between 65 and 94% control depending on species. So common lamb's quarters was a bit lower, around 65%, but all of our other species were close to 90 or greater, so really showing high levels of control. And this is all pictures from some of the Petri plate assays. We did see some interesting things with common lamb's quarters, which we get into in the paper, where we were seeing individual like seed fragments actually germinating and growing either hypocolor or radical, which is just a kind of like a little interesting aside. Once we had our optimized settings, we wanted to try and get as close as we could to an actual harvest. So we took all of those weed seeds, mixed them in a bag with a bunch of potato biomass, ran it through the vine crusher, and then grew them out in field soil. And here we've got a picture of the processed one on your right and the unprocessed on your left as our control. And we saw that for our small seeded species, we didn't really get a significant level of control. It wasn't that we didn't get any control, we were fighting a bit of variability there. But when we compare it to our other species with those larger seeds, we got between 37 and 83% control. So that kind of highlights the importance of, we think, seed size for harvest weed seed or for vine crusher efficacy. So we've demonstrated successful harvest weed seed control of problematic species in potato production and the first instance of harvest weed seed control for a horticulture crop. But obviously it's not as effective as these rolling cage mill systems, so we have an opportunity to improve some efficacy in the efficiency of the vine crushing. And the last thing I just want to say, this isn't the end of the story. We're still working on this and moving into field testing now to see whether or not this actually works in field. And so we've got an experiment running the last couple of years. This is just what it looks like at harvest. So you can see that plant material exiting the back of the vine crusher. And here we've got an experiment where we took a known number of seeds and known viability and ran it through the vine crusher at harvest. And that's what you can, if you see these little flags and bags throughout the field. So those have weed seeds in them that we're monitoring throughout the winter. And then we layered on another cultural practice, seeding of fall cover crop post-harvest, to see if there was any kind of synergy between these two practices. So with that, that's all I have to talk about. I'd encourage you to read our paper if you want to know more. And if I have any time, I'd obviously thank you to all of my co-authors here, Ashley Nicole McDonald, Laura Anderson, Scott White, and Christine Nerona. And I'd be happy to take any questions if there's any time. Thanks.
Thank you, Andrew. Yep, plenty of time for questions. I'll just, if you got a question, pop your hand up or throw it into the chat for Andrew. So I want to just to clarify on that last little bit about the continuing experiment. So the bags of weed seeds, maybe I missed it. Did they go through the vine crusher?
Yeah, sorry. So yeah, we took a known number of weed seeds of I can't remember how many species. I think there's 5. Put those in the bags. We know the viability of those, the initial viability, and then process those. And so we've done this now 2 years. The first year we attempted to just drop the seed bags as we were going through the harvest. But I think we ended up with a couple of issues. So then we ended up just processing them prior to putting them harvest, and then just laid them in after.
Okay. Gotcha. And so then they'll sit on the surface uncovered, right? Essentially in those bags?
They're kind of semi-covered. So just close to the amount of soil that would have been dumped back on it post-harvest. And then we did take soil cores as well to look at the seed bank. So we have pre-harvest soil cores, and then we'll do soil cores again in the spring to see if there's any change in the full seed bank within that top layer.
Excellent. So a very pertinent question here from Dr. Bay. How much would it cost to build a potato vine crusher?
So this is the original plans for this. It was originally funded through the pesticide risk reduction program from the Pest Management Center. So the plans are freely available. It could be anybody can do this. There's no IP protecting anything or preventing you from building your own. It is literally just two metal rollers, each hooked up to a motor, and then held together with springs. I am confident any farmer could build one of these for a couple of $1,000s.
Does it take a lot of horsepower to drive like the cage mills do?
No, so it was running. It is hydraulically powered, and it's just running off the back of the harvester. Just the hydraulics on the harvester. Yeah, and it has no. Yeah, I think the number I heard from Brienne last time, I think was around 60 horsepower draw to run one of those integrated seed destructors.
Yeah.
Whereas I don't think this would even register.
Cool. So from Jeanette here, are any potato processing companies like McCain's interested in or supporting this work given their interest in regenerative or sustainable potato production?
So we haven't had any, we haven't had either McCain's or Cavendish approach us. We have had the local industry though. So our local, the PI Potato Board is quite interested. They'd like to, or have talked to us about actually getting it out in producers' fields. I did the rounds, I think, a year ago on all the potato agronomy podcasts. So you can check out Tuber Talk and Spud Chat. So those are both two potato industry focused podcasts. And they're both interested. So we do have, although we don't have any of the processor buy-in, we do have industry buy-in.
You get the best names for potato
Yeah.
podcasts, don't you? Really? Like
Tuber Talk is pretty good.
Oh, that's great. Any other questions, folks, from the audience? We still have plenty of time for Andrew, so. Okay, well, with that, we'll say thank you to Andrew, and again, thank you to all of our speakers, to Eric and Sean. Really appreciate their contributions, and then to the many people who are in the audience who I know contributed to the special issue. Thank you very much. We appreciate it, and we appreciate our partnership with Canadian Science Publishing. Again, CJPS is our sponsoring journal for the Canadian Weed Science Society, and we really like to encourage that relationship and maintain a good relationship with CSP. So I'm going to hand this back over to, I believe, to Dr. Nurse or to Jeanette, and we're going to proceed with the AGM for Canadian Weed Science Society.
Okay. Thank you, Eric. Thank you again to all of the speakers. Those were some great talks. We really appreciated those talks, and I want to encourage everybody again, check out the special issue. There's, I believe, 10 papers in that special issue, so there's lots more very interesting research to be seen by going there. We will put that link on our website so that you can find it easier. And also thank you, Eric Page, for organizing these talks for this meeting, and to the special issue editors, Jachul Bay, Eric Page, and Charles Geddes for organizing this issue for the Society. Okay. So moving on, I'm going to hand it over to Rory Degenhardt, who's going to give a little bit of a memorial for one of our recently deceased Society members. Are you in the room, Rory?
There we go. Can you hear me now?
Yeah.
Sorry about that. Try sharing my screen. And can you do that again? Can you see my screen if I do that? Can you see my presentation? Or am I not sharing?
I'm not sure.
Currently don't see it.
Okay, I'll try again. Share screen one. Okay, now do you see it?
Yes.
Okay, perfect. Well, good morning, good afternoon, CWSS members. I have the special honor today of paying tribute to a legend in our industry. Dr. Linda Hall passed away on September 17th of this year, following a short and stoic battle with cancer. Linda was a CWSS member and fellow, past president, graduate student supervisor, presenter, session chair, and one of the most recognizable and consistent faces to our society over the great many years she contributed to our success. Linda received her PhD from the University of Saskatchewan and completed a postdoctoral fellowship in Australia. She worked for Alberta Agriculture and then settled at the U of A. She retired in 2019 as a full professor from the U of A. In her time at the university, she supervised numerous grad students, both MSc and PhD, and was also active on supervisory committees or as a co-supervisor for many more. She was a gifted and sought-after extension speaker due to her ability to tell a story, connect with her audience, and convey complicated scientific principles in terms that anyone could understand. Linda was a scientist who was curious about everything. This led her research program to cover everything from weed biology, herbicide management, and transgenic biosafety to agronomics like plant growth regulators and soil fertility. Dr. Hall's research program was world-renowned, and her contributions to the field of weed science, herbicide resistance, and transgenic biosafety helped put Western Canada on the map and helped countless growers to manage their operations more sustainably. She was a bundle of energy, and that energy didn't slow down after she retired. Instead, she channeled it into artistic and active pursuits, including gardening, hiking, quilting, and kayaking. A few weeks ago, a group of Linda's graduate students all got together to celebrate Linda at a virtual connection. It was a wonderful evening filled with stories, much laughter, and more than a few tears. The memories we shared followed many similar themes. She recognized the potential in all of us and devoted herself to helping us realize that potential. Her measure of success was our measure of success. Her bar was our bar. And maybe those lines blurred from time to time, but that made it all the more fun. It was clear that she was more than a supervisor for all of us. She was a teacher, a life coach, a role model, a mentor, a consummate professional, and a dear friend. I had the incredible honor of being Linda's first graduate student at the U of A. She set me on a path that has brought me tremendous joy and given me the opportunity to do things that I never thought possible. Linda forged her own path in everything she did, and I'm forever grateful her path intersected with mine. Our weedy world was made a better place by Linda, and her legacy lives on through the culture of our society, the scientific advancements that she led, and the members she collaborated with and supervised and/or inspired. Let's all take a moment to raise our virtual glasses in the air to toast Dr. Hall for her passion, her laughter, her mentorship, and her friendship. Cheers to Linda.
Thank you for that, Rory. That was a very nice remembrance for Linda. Okay, so I think we're gonna move into our annual general meeting now. So I'm gonna share my screen to bring up our presentation for the annual general meeting. I think we are a few members short of reaching quorum, but I'm gonna recommend that we move forward with doing the annual general meeting, and then Jeanette, myself, and Sean can figure out what we need to do as far as not meeting quorum to get some of the stuff passed at a later date. But since it looks like we have about 30 members here. We need 40 for quorum. So we're not that short. So we'll figure that out in the future. So I'm just going to share my screen here. Can everybody see my screen? Okay. So My computer's been acting up, so just bear with me. So as far as the agenda, we did provide the agenda ahead of time by email. And so essentially what we're going to do now is go through the president's report, the first vice president's report, and the second vice president's report. We'll then go through the treasurer's report, and then we'll finish off by talking about some of our upcoming meetings that we have for the society. As well as the minutes, they were provided by email, so hopefully everybody had a chance to review the minutes prior to the meeting. I guess, do we need to have a motion to accept the agenda in the minutes, Sean?
No, I don't think so, but we're not a quorum. We're just kind of going
Okay.
through the motions.
Okay. So, and there was no business arising from those minutes to deal with. So I'm going to hand it over to our president, Jeanette, now to take over the talk. I'll move the slides for you when you're ready.
Yep. I think we can advance to the next one. We'll jump right in. So most of you know, especially if you were in Winnipeg, I did take on an acting president role in October of 2023. This is a little bit out of the norm, typically when we don't have our November in-person meeting. The term has been extended to February 2025, but we are going to officially recognize the new board as of today, or when we can get approval for the motion. I do want to thank Harold Wright, who was acting president, for his leadership and guidance. I've told him this many times, but it was invaluable. And then also, without going into detail, just some personal reasons for making this decision. And I think you will have already noticed that Rob has been in a leadership role for this meeting here. And he's done a lot of work, and I really just want to extend a huge thank you. Again, I've told Rob, but he deserves a lot of recognition here as well. We also have asked if he feels healthy, and he's given us his guarantee, so no pressure, Rob. We also have a very fantastic board. I can't say enough good things about them. These are all volunteer positions. Everyone's really stepped up. So maybe just a bit of a plug here before we move to the next slide. If you are looking for some professional development, maybe just something a little bit different, and you are interested in a role on the board, raise your hand. We're always looking for people. So we can move to the next slide. Highly anticipated. Everyone's looking forward to the next meeting. And I think with good reason. David Clements and Victoria Brooks are working with the WSSA crew, and actually Rob as well, on the meeting taking place in February in Vancouver. And Jachul has promised sunny weather. I heard him say it when we joined the meeting. And then, not to be outdone, next up will be Ottawa, November 2nd to 7th, 2025. We'll be back on our regular schedule, and we have our chair, Wendy Absol, and then her committee as well, who have been putting things in place. And I think I might have it on another slide, but you'll see that. So mark your calendars. Board of Directors meetings were held in November 2023, March, April, and August, and an executive meeting was held in November of this year. It's pretty straightforward. Can move to the next slide. This year we did renew agreements for the Wild Oat Action Committee. Thanks, Brianne, who's not on the call, I don't think. And again, the Ottawa meeting, dates, hotel contracts, etc., have all been signed off on. The travel, the Student Travel Enrichment Award, I'll get into maybe some of the specifics on what changed in the MOP there, but it was awarded for the first time this year in 2023-2024. Next slide. Strategic planning, that is a big to-do. I don't think I need to steal Rob's thunder, but that will be, he's already actually set up a in-person meeting adjacent to the upcoming meeting in February, and that will kick off the full review planned for 2025. And next, I guess some of the other things, executive assistant is being reposted. So we did have some really good candidates that just didn't work out. And so we do have some interest that came in after the posting was taken down. So I do think there are some opportunities here, but also if you know anyone who might be interested in this role, it will be posted relatively soon, and we will be looking to fill that ASAP. Some other things that are always on the radar and will be a big part of Rob's upcoming year. Always looking to encourage students to be members and active members within the Canadian Weed Science Society. They are the future of the society, and so it's really important that we get them involved. The industry update section, there has been a request to consider an industry update section in our conferences, our meetings. And so actually our industry crew, they are actually pulling together a proposal at this point. So they have brought this forward to the board and did do some survey work as well. And strategic plan, I already alluded to that, so I don't think I need to go over that again. And I think there's one more slide. Actually, I was going to say this is it, but there is one more important slide here. So big shout out to Sean. He's our governance. He's our member that takes care of that, and he does it very well. And so he does updates to the MOP. So some of the things that did change, as alluded to, we did have the Travel Enrichment Award. And it is a very new award, so there are maybe some growing pains with this one. And so as a board, we did recognize that it was probably more effective to have 3 awards worth $5,000. That would go further in this case. So that was changed. And then it was also brought to our attention that some of the eligibility requirements weren't in line with some of the rest of our student awards. And so we have made changes there as a board to allow Canadian citizens studying graduate programs outside of Canada to also be considered for this award. So thank you, Sean, for taking care of those. And unless there's any questions, that is the President's report.
Being no questions, I guess I'll move on to the first speaker. Thank you, Jeanette.
Actually, Jeanette, just one quick question. I noticed the dates for the November meeting are earlier than in many of our historical meetings. What was the rationale for the earlier timing for the Ottawa meeting in 26 or sorry, in 25?
It came down to hotel preference and availability. And there really is, well, I guess Sean can speak to it better. There is a bit of a timeline that we have to fall within to have our AGM, but those dates fall within it. And Wendy also, on behalf of the membership committee, they do send out surveys after all of our meetings. And one of the big questions is, what timing is generally best for the meetings. And in general, November, not a specific week, or February are listed. And so it was just decided that this still falls within the timeline. And so it was decided to go ahead so that they could get the hotel of choice in this case.
Okay, thanks, Jeanette.
Thanks, Rory.
Are there any additional questions? Okay, so I'll move on with the first VP report. Oh. One of my roles as first VP is to look after the program sections for the annual meeting. So I'll put up on the next slide, I'll put up a chart of our program chairs and vice chairs, but essentially these positions are for 3-year terms. And the chair solicits presentations, posters for the section, organizes the presentations once they're received, and then coordinates with other chairs if needed. And then there's a vice chair behind them to take over once that term is over. So this is who we currently have assigned to these sections. So some of these, you will notice that we are missing vice chairs for those sections. So we would appreciate volunteers coming up and helping out in those areas if you feel that you have some expertise in that area and would help out. And I'm sure the current chair of those sessions would appreciate somebody coming in behind them to take over. So we will be looking for some volunteers for some of those sections. For the upcoming meeting in February, we did ask that a lot of those chairs or co-chairs help to co-chair some of the WSSA sections. So we won't be having the traditional CWSS sections at the meeting. We will be going by the WSSA sections. So we were lucky enough to fill a lot of those slots. Again, we do have a few empty ones. So if there are members out there that feel that they would like to volunteer and help out, we would be very appreciative to fill some of those slots for those sections that are still blank. So just let us know if you're able to do that for us. The next thing that we are part of as a society is the Plant Canada meeting. So we are one of the member societies. And we're lucky enough to have Andrew Mackenzie-Gopsel and Rory Degenhardt to be our CWSS reps for that meeting. So I guess they had 2 meetings for members this year, July 7th, October 16th, where they discussed a number of things, reviewing incoming and outgoing membership, new postdoc presentation award. They reviewed Plant Canada 2024, society updates. They're in a good financial position as a society, and they looked at the Global Plant Health Council membership. In addition to that, they did have a symposium looking at plants adapting to a changing world with a keynote speaker, Dr. Sylviane Charbois from Dalhousie. They also had some plenary talks included there where some of our members were giving talks, specifically Doctors Sarah Martin and Eric Patterson, looking at emerging technologies to enhance crop production in a changing environment. Overall, though, the consensus is that we did have very low participation from our CWSS members at that meeting, and we would like to see that increase in future years. So we want to look at how we can do that. Because at the most recent meeting, we had 7 abstracts from CWSS, and 3 of those were from the same author. So we want to increase that participation going forward. And one of those ways that we might consider is looking at making it our annual meeting when in the year that they're holding their meeting. I think that's what all the other member societies do for that meeting. And it may be something that we want to consider doing for our meeting as well. And that may get more participation from our members at the Plant Canada meeting. With respect to the publication director report, the publication director is our liaison with the journal, Canadian Journal of Plant Science. They also look after our website. So as far as with the Canadian Journal of Plant Science, there were 2 published manuscripts this past year. with a weed science focus. They were short communications. So we definitely want to encourage our membership to publish in the Canadian Journal of Plant Science so we can get those numbers up. And then as you saw earlier in our meeting, there was a special issue, Forging New Paths to Manage Herbicide-Resistant Weeds. There was 10 articles in there, along with 2 reviews. And again, the link is published on the website. And you can go directly to the Canadian Journal of Plant Science website and find that link as well. We do have the 2 Canadian biology series. We have the Biology of Canadian Weeds and the Biology of Invasive Alien Plants. And G-Tool oversees this committee as well with members David Clements and Sarah Martin. Unfortunately, for the past year, we did not have any manuscripts that were published for either of those series. So there is a long list of assigned and available accounts that are available on the website. So if you are willing to put the work in to do one of these accounts, it would be very appreciated, and you can check out those lists on the website to see if there's an account that you would be the appropriate author for. And just either let G-Tool Bay know that you're interested, or you can contact our subject editor for the journal, which is Dr. Ed Page. So the committee is planning to have another meeting probably early in the year. So if you have any suggestions for them, just let them know. We will be presenting the CJPS Outstanding Weed Science Paper Award again this year, and that'll be presented at the February meeting in Vancouver. And the committee members there it's being chaired by G-Tool and Eric Page, Sean Sharp, and Scott White are committee members. And then as far as the website, I hope everybody has a chance to go see the website. There have been some major changes and upgrades to the website. I think G-Tool's done a great job updating it, and he continues to do general maintenance with it as needed. Brianne Tideman is the chair of the Resistant Wild Oats Action Committee, and she sent a report on what that committee has been looking at over the past year. So they do have a number of great infographics and videos on our website. If you go to the website and you go to the materials page, you'll be able to find the Wild Oat Action Committee section there, and you can take a look at all of this great information. They did have interim funding for 23-24, which has now expired. And unfortunately, the proposal that they put forward was not approved for funding. So I think they're going to continue their extension work, doing infographics, fact sheets, and other things, but they are going to be looking for future sources of funding so that they can allow this activity to go on in the long term, and so that their project manager can be hired full time. So they are working on a new plan to move forward. And so I'll open it up to any questions or comments for that report. Does anybody have any questions? Okay, if there's no questions, then I'll hand it over to Andrew, who is our second VP.
Perfect. Thanks, Rob. Okay. So start with the WSSA report from Rob Nurse. So the upcoming meeting is a joint meeting with the WSSA, which we've already talked about, coming up in February in Vancouver. And the LAC members of that is, already mentioned already, but David Clemens, Victoria Brooks, Brianne Tideman, and Alita Janmet are all on the CWSS side of things. And from the WSSA, we have Hillary Sandler, Greg Dahl, Ian Burke, and Eric Gustafson. We've got 4 symposia that have been decided upon. The Endangered Species Act Committee of the WSSA, the WSSA Publications Committee, Not Your Old Professor's Journals, experience and insights from recently funded weed scientists in NEFA's competitive grants program, and Weed Biology and Ecology Committee, the contributions of genomics to non-target site resistance knowledge and management. So yeah, an exciting meeting coming up. I hope to see you all there. Moving on to scholarships and awards. This is from Leo Galinda Gonzalez. And the committee of that is Sean Sharp, Dilshan Benaragama, and Chris Budd. And probably one of the important newest things of that it would be the Travel Enrichment Award, where we discussed the changes to the MOP during the board meeting and discussed them today about including Canadian citizens studying at international students to be be eligible for the award as well. There's some discussion around timelines just to make sure facilitate the students actually traveling to their institution to be present at the actual at the meeting. So just some discussion around timelines there. And then contacted the two awardees from last year, Isabel Aiken and Marinda de Geer, to inquire if they would be completing their lab visits or returning the money. And both have completed their trip and will be sharing a short video of their experiences. And there was a call for hosts shared with members on August 27th and applications to the award on October 1st. I believe that's probably still open now. General awards, there was a call for application was sent out by Tasha and Jitchell on October 10th, and shared with membership and on the webpage. And so far received one submission for the Fellow Award and one for the Excellence in Weed Science Research Award. Moving on to the membership committee, this is chaired by Wendy Aspel with Rory Dagenhart, Will Kramer, Jonathan Rossett, Mary Jose Simar, Brianne Tybman, and Harold Wright. For 2024, they delivered and reported on the CWSS SCM member survey and meeting feedback and compiled suggestions from members for use by board and local arrangements committees. And so we've heard just a couple of comments on that already today. Ongoing discussions on how to increase response to surveys. I mean, that's always a challenge. And there's 5 winners awarded maple treats for completing that survey. Finalizing the criteria for the CWSS Early Career Professional Award to be conferred in honor of the CWSS fellows and members that are no longer with us. And there's an ongoing inventory of recent additions to the CWSS archives, and a list of items available is on our website. And there's been some discussions on weed science capacity concerns across the country, just with continued consolidation, retirement, positions not being backfilled. And there was a proposal submitted to the board from membership just to advocate for more weed science positions within Canada. For 2025-26 planned committee activities, design, deliver, and report on the joint CWSS at WSSA meeting feedback and general membership survey, and then publish the CWSS membership review from those surveys. I'd like to update the CWSS SCM outreach materials for the purpose of the membership benefits, and pursue various activities as per the board direction. And we really do appreciate everyone taking the time to fill out those surveys. They do get read, we do review them, and the information that's in there does get used to try and better serve our membership. Oh, I think Rob dropped off. And I lost I don't have a copy of the presentation handy.
I can pull it up, but I'm wondering if Chul maybe has.
No, I don't have one here.
Okay. Might just take me a minute. I'll just be quick here.
Sorry, everyone. It seems my computer decided to drop the ball.
Not a problem.
Let's see if I can get back to the presentation. I believe you were on this slide.
Yeah, we just finished the membership report. So, okay, nominations and elections. So this is chaired by Harold Wright with members at large. And so committee meetings were held on June 27th and in October 28th, I assume. And call for nominations went out on July 17th and closed on September 27th. And yeah, all positions are filled by acclamation. So regulatory rep. Oh, okay, sorry. So we need a regulatory rep. Wendy Aspel is stepping down, but willing to stand for re-election. Okay. All right, and then second VP. So I'm moving to first VP, and Charles is taking over for me. And then member at large West Dilshan is outcoming, and Ethan Berthollet is taking over. And the communications committee. So this is chaired by Tasha Wright, and they met once this year to discuss potential newsletter topics, published and distributed 7 monthly newsletters, and included information applicable weed science events from outside the CWSS to try to broaden the network as they just try and search for more weed science topics. They were active on Twitter, Facebook, and LinkedIn, all under the CWSS banner. And going forward, they want to work to find ways to increase participation from our graduate students in the society, things like defense announcements, where they are now, that type of thing. Encouraging interviews with retired or retiring weed scientists from across the country, just kind of a career perspective and retrospective, and then improve interaction and input from membership on newsletter content. And Tash is always looking for any kind of ideas you may have, any feedback, topics. Yeah, I'm sure she's open to anything that any suggestions you may have for her. Okay, and then I think this is the last one, the graduate student representative report. And so that's Will Kramer. Conducted a graduate student specific feedback survey, compiled information on the grad student experience at the 2023 annual conference and what opportunities should be offered in the future. In terms of upcoming events, they have a grad student mixer planned for the joint conference and a seminar at the upcoming conference titled The Graduate Student Experience Perspectives from Government, Academia, and Industry in North America. And then lastly, they're working on organizing a bioinformatics workshop with the WSSA graduate student representatives, with more information to come. That's all I have.
So I guess because we don't have quorum, John, do we need to put forward a motion or?
No, I think we'll have to sort that out later.
Okay. So we'll move on. Is there any questions about the second VP reports? Okay, so I guess we'll move on to the treasurer's report. I don't see, Deanna said she may not be able to make the meeting, so, and I don't see her in the chat. Unless I'm missing her. So I think I'm going to present this report in her absence. So just bear with me because I didn't do all of the accounting for this. I'm just going to be reporting on the numbers that she provided. So don't hold me to anything. As far as sponsorship, Megan Lang and Tyler Gullin were responsible for sponsorship for our meetings. They have updated the sponsor contacts in 2024, and they're anticipating that there'll be 13 national sponsors for 2024 to a total of approximately $60,000. So again, we thank all of our sponsors. We couldn't do it without their generous support. This is a snapshot of our sponsors. We really appreciate all that they do for the society and all of the support that they give. We wouldn't be able to hold our meetings without our sponsors. So we thank them again. As far as the overview of the financial statements, I guess I should mention, I believe G-Tool did put the financial statements, a link to them in the chat. So if you want to take a look at those or download those, The link to the financial statements is in the chat. They were submitted to BDO Dunwoody for draft review. We have received it back with a final copy pending. As of the date, September 30th, 2024, we had about $239,000 with cash on hand at BMO. We had $0 in our Scotiabank account because we were switching over from Scotiabank completely to BMO. At Scotiabank is where we had our credit card and we had a collateral GIC account. Those were both closed in 2024 and they were transitioned to a BMO credit card. So our investments are now at BMO and there's about $111,000 at BMO. 41,000 is in a GIC. About 65,000 is in mutual funds. And then we have a $5,000 GIC, which acts as collateral for a credit card from BMO. Overall, we always look to meet 2 years of operating expenses, which is $113,000. And so according to these numbers at this time, with our cash on hand, we are meeting that objective of having 2 years of operating expenses. Just going into the numbers a little bit more, this is just a chart of the last several years. She has highlighted 24, 25 there as far as what our most common expenses are for the society. We did have an additional amount in there to hire a facilitator to engage in looking at our strategic plan and our long-term objectives. So that is worked into the estimates to be able to hire a facilitator. We may choose in the future not to do that, but we just wanted to have that earmarked so that we have the ability to do that. Also, as far as our subcontractors, we do have in there the $26,000 for the executive assistant. But that, again, that competition is still ongoing to fill that position. So we do have that earmarked, but the position is currently vacant. And then the other things are just things like insurance for the board of directors, travel for the executive assistant, office supplies, hotel deposits, and other miscellaneous things, including bank fees. And some of the other, so that subtotal on the previous page came to $51,540. Some of the other expenses that we have worked in there is funding for the Resistant Wild Oats Action Committee from our society in the amount of $15,000, as well as what we talked about, 3 $5,000 rewards for the travel enrichment program, which would come to a total of $15,000. So our total budget that we were looking at was $81,540. If we look at the income from the annual meetings, we look back at Halifax, we had membership income from dues and everything about $14,250. And the AGM profited about $31,071. Versus in Winnipeg, we had about the same amount coming in from membership dues, $14,400. But our AGM profited close to $71,000. And it was just noted by Deanna that this difference in the numbers was due to the level of charges by the hotel, what they charge for rooms, food, etc. So that's why there is a difference between those two meetings, when it appears that membership attendance was about the same. In 23-24, we did have 96 members that attended. So that financial report was submitted to BDO and the review was received. And based on their review, nothing came to their attention that they red flagged. So they did accept the report as submitted. Again, we're still waiting on the final review from them back. But that's how it stands as far as the BDO review. So at some point in the future, we are going to have to get the membership to approve the treasurer's report as presented. We also need to officially appoint BDO Dunwoody as our auditors for our future financial report. So we'll have to work with Sean to make sure that we get that done in the official manner. Before I move on, is there any questions for the financial report? Okay. As far as the resolutions committee, this was headed up by Martin and Dilshon. Prior to the meeting, I contacted them asking if there was any resolutions that had been submitted in 2024, and they informed me that there had not been any, and I haven't seen any leading up to the meeting. So I think we can leave it as no resolutions have been submitted to this meeting for discussion. So just before we get to the end of the meeting, we'd just like to take another jab at advertising our upcoming meeting in Vancouver, February 24th to 27th. It is going to be at the Sheraton Vancouver Wall Center. The room block is now open. There is a link on the WSSA and potentially the CWSS website to be able to go and book a room at the hotel. We're asking that presentation titles are submitted by December 2nd. That site is open. The registration site is open for CWSS members to go register, and early bird registration ends January 18th. So you have till then to get in to get the early bird rates. As far as highlights of the meeting, we are going to have a keynote address by our member of our society, Indigenous scholar Jennifer Grants. We're going to have 4 symposium, which Andrew mentioned earlier. We still are going to have as a society our CWSS student presentation contest. We feel very strongly that that's a very important part of our society, and we want to continue that on. And there'll be 3 field tours that attendees can take part in. There'll be a Granville Island tour, a craft beer tour, and a harbor tour, as well as a weed science improv show. So we really encourage you to get your titles in and register for the meeting. It's going to be a great meeting in Vancouver. And then I'll give a little bit of a plug for the meeting coming back to Ontario. It's going to be November 2nd to 7th. 2025, and it's going to be at the Hilton Lac Lemay in Gatineau, Quebec. And it was just noted that for travelers coming in that are going to be coming into this meeting, that the Ottawa airport is the closest airport to the hotel. So when you're coming in to fly in, make arrangements to fly into Ottawa. So keep your eyes out for emails and social media announcements for this meeting. That'll be coming over the future months, as well as stuff that's put up on our website.
Hey, Rob.
Yep. Yes,
This is Hillary. Can I interject one or two things about our joint meeting?
absolutely.
So for the room blocks, I don't know how people will be traveling, but we are currently working with the hotel to get Thursday night at the same room rate as the rest of the meeting. So if people know that they're going to stay over Thursday night, I guess I would encourage you to grab up your hotel as soon as possible because I think we have about 20 nights for sure. But we are working with the hotel. So if you have any concerns, please just reach out to me. But that is our intent. And most likely don't tell anybody I said this, but we'll probably extend the title submission for probably at least a week. But we absolutely really encourage you to go ahead and get your titles in as soon as we can. We were a little bit late this year because we ventured into a new submission platform with a new company. So it took us a little while to kind of get our act together. So, but again, if you have any questions or anything, please feel free to, reach out to, obviously Rob or Dave, or reach out to me. I'd be happy to try to help you guys. And we're looking forward to it. So thanks.
Yeah, perfect. Thank you, Hillary. Is there any other questions or comments about the meeting before we move on?
Yeah, this is Greg. I also would like to invite you to come on for you guys, come on over to Vancouver. And for those of us that are in the United States, we're going to come on up. I think we'll have a wonderful time, and I thank the program planning committee for putting together a great meeting. So come on over. We'll have us a good time.
I agree. Yeah. So everybody get registered for that meeting.
And the program booklet should be up on the website, I would think, probably by the end of this week. But as Rob mentioned, some of the tours are actually going to be Thursday afternoon, which is why I made the mention about people potentially spending the night on Thursday.
That's a good point. Yeah. Okay. So I think we'll move into the last part of the meeting, and I'm going to hand it back over to Jeanette to officially do it. Or I guess because we don't have quorum, it'll be unofficially at this point.
I was going to say, it's also a little bit anticlimactic over webinar. Don't get to hand over the gavel, but we will figure that out. I think Rob has definitely demonstrated his leadership here. So society's moving into good hands moving forward here. So Rob, welcome aboard, Vice President.
Thank you, Jeanette.
We also have our new board members, as indicated here. I don't even know if Charles is on. And Ethan Berthollet as well, so our member at large for the West. Again, if you are interested, this is a great group of people to work for. Of course, we're acknowledging new board members, but there's a fantastic crew here in the background, many who you've heard from or saw their faces on the screen today, but not everybody. Again, just a great group, and yeah, looking forward to working with you, Rob.
And I just want to say thank you, Jeanette, for the time you've spent on the board and as moving up the ladder to the president, and thank you for serving as president. It's been a great experience working with you, and I look forward to working with you as past president in the role as past president. And I also take the opportunity to thank Harold Wright, who's also moving off of the board as the current past president, for all of the years and service that he put onto the board and the experience that he brought. So thank you to everyone. So I look forward to moving forward over the next year. So we'll end it there with seeing everybody at the next meeting. Thank you, everyone.
Thank you, Rob.
Yep. Thank you.
Thanks, Rob.
Bye.
Thanks, everybody.
Yeah.
Great job. I see you.